I'll just go live on YouTube also, because we have quite a few of our viewers on YouTube now.
Then one thing, what is the, see, suppose I've used very similar slides in other talks, which also happen to be on YouTube. That is, I don't know, that's sort of reasonably okay, I suppose. Many of this will be, some of the data slides, some of this, all these have been used in other talks also.
Okay. No, if you wish, if you like,
no, I have no problem. My own,
okay.
These are my own talks, so I don't see why they should be. That obviously, like when we are giving popular talks, not all of it is totally new or something like that. So that way.
Yeah, sure. Yeah, I think we can start now. So I'll start with the introduction. Okay. Hello everyone. Welcome to the Department of Chemistry, IIT Delhi. Wish you all a very happy New Year, and thank you very much for joining this webinar today. So my name is Soumik Siddhanta, and I'm the webinar facilitator and an assistant professor at the Department of Chemistry, IIT Delhi. And we are very pleased to bring you this content today, which is the part of the online Pratidhwani series that the department is organizing. So the webinar series is sponsored by Author Cafe. Author Cafe is an online platform that aids with writing, collecting, and organizing, collaborating, and publishing your research content through features and integrations with products like Mendeley, Zotero, Orchid, Crossref, and so on. Think it like Google Docs, specially designed and developed for the academic community, and much more. So today we are thrilled to have Professor Subrata Sinha from the All India Institute of Medical Sciences, New Delhi. So before we start, a couple of technical details. Please do not unmute yourself during the talk. If you have any questions, please type them in the chat box, and we will try to get them, at least some of them, through to the speaker after the talk. So Professor Sinha is currently the professor and head of the Department of Chemistry at AIIMS, New Delhi, and also a J.C. Bose fellow. Decorated by several research felicitations at national and international levels, Professor Sinha's research contributions have been well respected in the field. Early in his career, Professor Sinha graduated from India's premier institution, the AIIMS at New Delhi. He was awarded MBBS in 1980 and MD in biochemistry in 1983 from AIIMS, and PhD in 1989 from the MRC Toxicology Unit, Carshalton, UK. Professor Sinha was an ICMR Talent Search Scheme Fellow at the Department of Chemistry, AIIMS, in 1984, and at National Institute of Immunology from 1987 to 88. He was a Smith and Nephew Fellow and non-clinical scientist at the MRC Toxicology Unit in UK. He joined the faculty of the Department of Biochemistry at AIIMS in 1988 and was appointed as professor since 1998 and head of the department from 2003 to 2010. Professor Sinha is the recipient of several awards and honors, including Fellow of Indian Academy of Neurosciences, Fellow of National Academy of Medical Sciences, Fellow of the Indian National Science Academy, Fellow of National Academy of Sciences, Fellow of the Indian Academy of Sciences, member of Guha Research Conference. He was also awarded ICMR Senior Biomedical Fellowship for visit to CBER FDA in USA in 2008. And he was also awarded the prestigious Swarn Jayanti Fellowship of the Department of Science and Technology, New Delhi in 1998. So Professor Subrata Sinha served as the director of National Brain Research Center, Manesar, from 2010 till 2017. He has been working on the molecular and cell biology of glial tumors and has made several important contributions in understanding of this fatal brain disease. His research contributions include understanding the molecular biology of glial tumor progression, studying genomic instabilities that may contribute to alterations in tumor phenotype, which in turn may confer drug resistance, study of cellular responses to stress, including hypoxia, transcriptional gene silencing, and its implications in tumor biology and therapeutics. He is also working on the development of recombinant antibody methods for potential use in oncotherapeutics. A very warm welcome and over to you, Professor Sinha.
Hello, good afternoon, and thank you, Shomik, for this very kind and generous introduction. And I am grateful that IIT Delhi Pratidhwani has invited me to give this talk. And I will be telling you, actually, I'm not from chemistry, I'm from biochemistry. And there is a whole lot aspects of chemistry that fundamental chemistry that I sort of fear and dread also. But having said that, I would like to talk about dyslexia. And this is an interest which I developed after I joined National Brain Research Center uh, from AIIMS, and then worked there for 7 years and before coming back to AIIMS. So what are we doing? What is dyslexia? So I'm going to talk about how study of dyslexia can take you from phenotype to genes and how it can help you initially not also to understand the biology of the disease, but before that it can help you understand kind of normal cell biology and functions. So also to remind you that a lot of my work, in fact, bulk of what I'll be saying was actually done at National Brain Research Center. This work is continuing after I'm back to AIIMS. And this, as you know, is a beautiful DBT institution set up for the study of biology. And these are the collaborating labs that have worked in it. National Brain Research Center, my lab, Dr. Nandini's lab, Pankaj Seth's lab. And from IGIB, we have a collaboration with Mitali Mukherjee and Dr. Muhammad Farooq. So cognitive and neuroimaging studies on dyslexia were initiated at that time by Nandini. And then we studied the familial and sporadic dyslexias that she had been also studying from a cognitive point of view. And all the slides relating to some basic aspects of assessment and neuroimaging are borrowed from. Oops. So some pictures are not coming up. So this is something which is interesting about reading. You know, reading is a particularly human function. If you look upon it, no other animal so far in its natural environment actually reads or has any equivalent of reading. Vocalization, yes. So spoken language is a biological specialization, but written language is a cultural invention. Spoken language is learned, is a natural thing. You know, vocalizations progress into more and more sophisticated spoken language. Reading is a learned behavior. But yet, and this learning is very, very essential because we need fluent reading skills for success in today's world. And so the brain has to learn to read. So it's a learned behavior. It's something that we learn while, you know, we sort of grow from, you know, toddlers to children to adults, and it gets more and more sophisticated with training. And how and what are the components involved in reading? There's a word and there's a meaning and there's a sound. So how do you, when you read, you obviously look at a symbol, you associate the symbol with a sound and you associate it with a meaning. Now, the flow can also be reversed. You may look upon a meaning and think of a word for it and then imagine the sound for it. So, we can, in a way, look at the word, get a meaning. When we read a sentence, what we are doing, we're assembling individual sounds and trying to read them and make a coherent view from that. The other thing is that, brain is something like a muscle. People say, why? You can train the brain, you can make it do tasks which is never done before. And, both in power, skill, sophistication, everything the brain is trained. Now, let's see something many of us have done in our childhood, learn to ride a bicycle. Now, initially, we can remember how clumsy we are. We've fallen down, skinned our knees, and basically, it's been a struggle. But slowly and slowly, you learn to cycle, and you can keep your balance, speed, coordination, and many of us can perform bizarre tricks in cycling. So all that is learned, and once you have learned, that whole process becomes automatic. So early experiences happen, underused connections. So it is because of brain connectivity. Learning is lot to do with brain connectivity in a very simplistic way. So if there are nerve connections which are underused, these are pruned. Repetitive use of connections leads to more connections, strengthening of connections, so-called, and learning. A lot of this mechanism is deciphered. A lot of it is still to be found out. Then there is no single area for learning. All There's a network of brain areas which are involved in learning. So this is something that we need to know, that of course lesion of even one of these areas can basically lead to a second, let me switch off. Lesion of any one of these areas can actually lead to difficulty in reading, but then all the areas together will be involved in the process of reading. And so what happens when the brain learns to read? New connections form, and this leads to learning. And there is this process that we all have heard of, many of us, which is called neuroplasticity. So there are two kinds of thoughts which have come into the brain. That one, there are different areas of the brain. You can see that people get stroke. People who get stroke cannot move one arm, one limb, because there's a particular localized stroke area which is affected. And some of them cannot speak, some of them cannot comprehend, some of them cannot write. Even these lesions affect because of stroke. So there are certain areas which are affected by reading. There are these fixed areas in the brain which affect reading or any other activity. Then there's this concept of plasticity, that depending, its plasticity is not absolute, but depending on the extent and the stage and age and everything else and training, other parts of the brain, to a greater and very often to a lesser extent also, learn how to take over the parts of the brain that have been damaged. And in neuroplasticity, learning to ride a bicycle is also a phenomenon of neuroplasticity, how the new circuits have developed. Same way, learning is also a manifestation of neuroplasticity. The other thing is that, reading is not very old. In evolutionary terms, it has it is about 5,000 years old, and possibly it has developed as a means to encode spoken language and to transmit the ideas from one place to another where the two people are not physically present in front of each other. And what does reading mean? Reading means that there is a set of symbols which are called graphemes, and these map to a set of sounds. So all writing systems, these were developed to encode this spoken language. Some reading research questions which are more cognitive in nature: how do children learn to read? Why don't some children learn to read well? What can we do about it? And so we come into a concept of skills or skills which are involved in the process of reading. And as I come to later, anything where there is difficulty in reading or in the absence of a globalized uh, defect in the neural processing, that would be very simplistically called dyslexia. So, in this test, children are tested for dyslexia in many ways. Naming an object, semantic fluency, the words, the meanings, vocabulary, how fluent is it, verbal fluency, rhyming, phoneme replacement, replacing one sound bit with another, word reading, non-word reading. These are various tests which the psychologists use to assess the process of reading. Naming pictures, you can do it in Hindi or in English. For example, rhyming, naam, kaam, but then the word meal does not really rhyme with naam and kaam. So, Hathi, Holi and Sati, obviously Hathi and Sati will rhyme, and so on. In English also, same way, tail rhyme, but boot doesn't rhyme, and so on. Okay. Phoneme replacement. Dog, if you replace D with an L, you get log. If you replace T with L with a T, you get a tip. So that way. Mahal, Chahal, so on. So I will not go so much into the process by which words can be read, or the various aspects of breaking up the process of reading. But I'll bring you to another aspect that recently, well, not recently, a few decades now, 2 decades at least, if not, functional magnetic resonance imaging is a key to identifying the areas of the brain activated during a specific task. And in this case, we can take reading. And people can be studied by fMRI. And what it does in the chemistry department seems redundant to essentially when the brain, regions of the brain perform a particular task, then the oxygen is consumed there and oxyhemoglobin becomes deoxyhemoglobin, and then the concentration of that in that specific location of deoxyhemoglobin to hemoglobin ratio can be measured by magnetic resonance. And why is it called functional magnetic resonance? Because it's not a static thing, it's a dynamic thing. You can actually see the function of different parts of the brain light up when this resonance, when a particular task is being performed. And as you can see, when people are reading in both Hindi and English, this is the brain from one side and the other side, and this is from one side and this is from the other side. You see, they all have a very similar areas of the brain lighting up. Chair, bawa, whatever, these are words, but notice the regions are very similar. So essentially, different parts of the brain will light up. That means they will be performing the task involved in reading. Okay, so now what is dyslexia? Dyslexia is reading disability. It is a neurodevelopmental disorder which essentially works to, it's a form of specific learning disability. It is difficulty in reading and writing despite normal intelligence and adequate education and opportunity. So somebody has never learned to read, illiterate person, child, unfortunately left out by development, that is not dyslexia. Somebody has an intellectual disability, that is also not dyslexia. Dyslexia, many of the dyslexic people are actually very gifted in many forms of, whether it will be spatial visualization, arts, even great theater personalities, and so on. So, but it is a specific learning disability. Something like dyscalculia. Dyscalculia means person cannot calculate. He may be wonderful at everything else. So it has a world prevalence of about 5 to 10%. These are rough figures. But then there is a biological basis to dyslexia, and people have done postmortems of brains of people who have been known to suffer from dyslexia, and they do find abnormal development, abnormal things indicative of abnormal neuronal migration. And these are psychometric assessment batteries which are involved in the process. I'll not go into it too much in detail. I've been told that we restrict to half an hour. Let me see. So when you also see that there is a spread of, it's a statistical assessment. You see, the greens are the controls, the reds are the people who are suffering from dyslexia. So there is a clear-cut statistical difference, but then the point is there is an overlap. This is when you see them on different scales. This is non-word reading. This is what? This is rhyming. This is phoneme replacement. Forget what RAN stands for. But anyway, another characteristic we check. So dyslexia, by the way, I'm not really much into cognition. Dyslexia in India also is an interesting case because many of our people, especially people from a higher socioeconomic status, are biliterate, and the heritability of dyslexia is very well known. This is the other thing that come to our talk, that there's a relative risk of developing dyslexia of about 40 to 60% with a child with an affected parents. And there have been many genetic studies which have identified several loci, DIX1 to DIX9, which are associated with dyslexia. And from these loci, there have been candidate genes, DIX1, C1, please remember this, will come later, DC2, ROBO1. These are all involved in basic processes of neuronal development or nerve migration. Dyslexia is a multi-gene process which which manifests because of gene-environment interactions. The other interesting thing is that, concentrate on this sentence, likely to be a basket of disorders with a broadly common phenotype. That means I read with difficulty, someone else also reads with difficulty. The end point is similar, but it's quite possible that both of us have had different neurodevelopmental defects which have led to it. So in a molecular sense, dyslexia is what they call a syndromic diagnosis, that you assess dyslexia from an endpoint behavioral scale. But actually in a molecular term, it is the two are two people might be very different. It's something like cancers. You see, in cancers these days, molecular sub classification of various types of cancers is becoming very common. The whole concept of classification of cancers has changed because you may have a high grade tumor in 5 people, and they may form 3 different baskets of molecular diseases. So the molecular pathway leading to that abnormal growth and invasion is different in A, B, and C. Same way, the molecular pathway in dyslexia can be different in A, B, and C. And in developmental disorders, this phenomenon is known as the endophenotype. Phenotype means manifestation. Endo means, inside basically you are able to classify them into different similar groups which within themselves have a consistency but differ from each other. So therefore comes the role of genetic analysis and complex phenotyping. So maybe they differ slightly from each other, we don't know. But broadly speaking, endpoint remains the same. So our subjects underwent different tests. I'll not go into that. And but then the other thing which I'd like to tell you is that we studied families. So Nandini was fortunate that the Maharashtra Dyslexia Association was very proactive, and they helped us to get what are known as multi-generational families, where you have many people being affected, but they also were endogamous. And why? What is endogamous? They belong essentially to the same caste, and for generations they were in a localized area marrying within the same caste. So, in USA, a lot of genetic studies are done on what are called a group of people who are Ashkenazi Jews. The reason is that they are very consciously endogamous. They have been marrying within themselves. So a lot of genetic diseases come up. And like suppose I and someone else, just I meet casually walking by on the roadside, our genetics, our DNA is sequenced. Lot of the differences between us, suppose I have a disease and he or she doesn't, but lot of differences in the DNA will happen just because of chance random changes. And the DNA changes attributable to the disease will not be very obvious. In an endogamous group, because they tend to intermarry, the differences between people which occur by chance between unrelated individuals are less. And therefore, there is a higher probability of picking up differences between the black and the white people who are affected person, not affected person. And so what we did, we studied the family, had a behavioral assessment, and then of course studied them vis-a-vis exome sequencing and NGS. This is another family. Here, in the previous one, what we found was that this was possibly an autosomal recessive inheritance, something like you see in sickle cell anemia, where one bad, we have two copies of a gene, but one bad gene is not sufficient to give the manifestation of disability. Here, in another family, there is a autosomal dominant. That means even if one gene is there, the person is very likely to get the disorder. So already, if the two genes which are predisposing in two families have different patterns, it is very, very likely that the genes are different, and the inheritance patterns points out to differences in the genes likely to be affected. This is another one. Inheritance in what are comorbidities. Now, dyslexia is associated with also comorbidity. That means a person who has dyslexia has a higher probability of also suffering from attention deficit hyperactivity disorder. So there are some people who basically, people with a block on the left have dyslexia. People with the block on the right are likely to have ADHD, and there's one set which has both. So this was an interesting phenomenon which we studied. What is the common neurodevelopmental basis for all of this? And to make a long story short, what we did was we did exome sequencing of the affected and unaffected people. I'll describe the first family in somewhat more detail. Got the SNPs which co-segregated with the disease and tried to then we validated them with various sequencing methods. And what we found out was in chromosome 5, there was this GT, dinucleotide insertion, present upstream of a gene. which is the BAS1, brain specific acidic protein 1. Now, this was a very interesting location. This is the BAS1, this is the coding gene, which is transcribed in this direction. In the opposite direction is transcribed one long non-coding RNA. And the first exon of the long non-coding RNA is common and overlapping with the promoter region of this BAS1 gene. And this defect, this genetic insertion, is located in a common region. And these are divergent LNC RNAs, what we are seeing. And that does function in neurodevelopment, which we know before, neural outgrowth, synapse formation. And we wanted to know whether these LNC RNAs also have a role in the regulation of this gene. Nowadays, it is, when we started off, it was new, and we've been publishing this work. But the developmental role of long non-coding RNAs has really come into fore. So many of these long non-coding RNAs actually help in, especially this divergent RNA is going to help in regulating the role of the coding gene. And this was important. I'll come to how we got to that fact. So we found that, okay, in different tissues, this is from a database, regions of the brain have this BAS long non-coding RNA, and these regions are also having the expression of the gene. In situ hybridization showed us that long non-coding RNA exists. And then we had a cellular model. So Dr. Pankaj Seth from the NBRC, he grows neuronal progenitor cells from aborted fetuses, kind of second trimester, early second trimester fetuses. And these are progenitor cells. And these are when you differentiate into neurons, and these are when you differentiate into astrocytes. And at the end point, what we saw is that both the long non-coding RNA and the progenitor cells have high expression of both the BAS1, which is black, and the long non-coding RNA, which is gray. And when in neurons it drops, and in astrocytes it drops even more. So we wanted to know whether one regulates the other. Did siRNA experiments found out that when you knock down the long non-coding RNA, you manage to knock down the coding gene also, which was not true when you did reverse experiments. When you knock down the coding gene, you don't affect the coding RNA. And then we wanted to study in vitro when the cells differentiate, how does it work? Simple thing, put siRNA, the HNPC was pushed towards cellular differentiation, both towards neurogenesis and to astrogliogenesis. See, brain has two kinds of cells. One is neurons and one is astrocytes. The neurons are the cells which have electrical connectivity. Well, so do astrocytes. But the astrocytes were supposed to be the supporting cells of the brain, but now they exist in dynamic equilibrium with the neurons, and they arise from a common progenitor cell. So it is possible to push a common progenitor cell towards glia. It is possible for it to push towards neurons. And this is how that differentiation was done in Dr. Pankaj Seth's lab in vitro, that you supplemented to media, supplemented with brain-derived neurotropic factor and platelet-derived growth factor to initiate neurogenesis, or just add serum and it becomes astrocytes. And you carry out this for 21 days, extract RNA at different days, and see what happens. So interestingly, this is the level which is constant in progenitors. But when you push a progenitor towards neuronal differentiation, there's a huge spurt in the long non-coding RNA as well as the coding. Doesn't happen in the astrocytes. It just falls down. It just disappears. Both the RNA as well as the coding. The protein does the same thing. These are neurons. It rises up and then it drops in neurons. So we wanted to study neuronal differentiation. So mind you, the study of the phenotype of dyslexia has led us to, the genetics has led us to a process of neuronal differentiation which wasn't known before. So what we do is we do knockdown experiments. We knock down the long non-coding RNA, see how the one marker of neuronal differentiation behaves, and see how one cell proliferation marker behaves. And then what do we get? When you knock down and make the cells differentiate, the neuronal differentiation marker's expression reduces. The proliferation marker increases, the cells proliferate both on day 1 as well as day 3. And also the neurite length. See, the progenitor cells don't have projections. These projections are known as neurites. You can imagine a picture of a neuron. So whatever these projections are at the rudimentary stages are known as neurites. So the neurite stage also decreases. So it means that this axis is also involved in neuronal differentiation. And we studied the mechanistics. I will not really go into detail, but we did find that the RNA of the divergent long non-coding RNA is actually bound to the gene. And here it acts as an enhancer. It doesn't act as a suppressor. Its binding to the RNA is important for the expression of the gene. We haven't gone further into molecular depth, but this much we are confident of, and we have described and published. And this is the model that we have seen. The gene is long. There's a huge intron, 58,000 bases, and the LNC RNA is 1 or 2 kilobases, 3 kilobases, sorry. And what it probably does, it binds to the two ends of the gene and helps in forming a loop which makes the gene, let me say, express more. We don't know how. Now, another very interesting thing came out, that these days a lot of data is available on the net. So we looked at the UCSC ChIP-seq data, and we find that there is a transcription factor called TCF12, which is bound to this region also. Now, what is TCF12? Well, we showed by a chip that it actually is bound to that region. So we were able to cross-link and pull down that region of DNA when we pull down the TCF12. And then what we do see is that when we do a BAS1 knockdown, again the neuron doesn't work, doesn't get differentiated. It's lower and the proliferation increases. When you do a TCF12, that means that transcription factor knockdown, also the same thing happens. A neuronal differentiation is slowed down and neuronal proliferation increases, and they have a common molecular pathway. When we did some gene arrays, we didn't do sequencing, but we did some gene arrays, and we find that there are many common genes. When you knock down either of them, either the long non-coding RNA or the coding gene or the transcription factor which arises from another region of the genome and by the coding gene, any of them, you get a very similar effect on certain key genes. Now, coming back to literature, what we found was that in our study, we had shown an endogamous family had a novel GT insertion in a novel region of dyslexia, which had not been shown before. Literature said that there is another DIX1 locus, which is on 15q, which is the long arm of chromosome 15, which is highly replicated in inherited dyslexia. And this one locus codes for TCF12. So what we see here is both these genetic loci affect a common pathway, which is a novel pathway, which is critical for neuronal differentiation. And so the same phenotype can result from the effects of different molecular pathways. So the outward manifestation is the same, but you can get different molecular reasons for actually having that manifestation. Coming towards the one family, the second family that I'm describing, there is another family which is called K25. Again, we did exome sequencing and validated with Sanger's, which has homozygous autosomal dominant disorder. And here in chromosome 17, there in chromosome 5, 5q31.3, different region of chromosome 5. That was short arm, this is long arm. There are 17 variations which are inherited in block. So there is one non-risk haplotype, there is a risk haplotype, and the red one is all of them are inherited together. This is also an interesting mechanistic study as to how co-inheritance occurs. But most of them are around the PCDH, protocadherin G alpha locus PCDH1 and its coding gene and its regulating genes. So what are cadherins? Cadherins are molecules on the cell surface. They cell-cell interaction between two cells. You see, when neurons develop, the long during brain development, the long neurons tend to move and migrate from one region to another. And neurons can be very long and thin, and they are sustained right through, because you have the cell body here, and the neuron is connecting with another neuron. Sometimes in the case of spinal neurons, it may be in meters, but in the brain, definitely it can be in centimeters. Of course, many of them are much less. And this migration, how does the neuron find its way to the right spot? The neuron finds its way to the right spot because of protein and molecular interactions with the other neurons which are developing. And they, in a way, it's like, I can't see, but I feel my way through a set of markers to reach a point. Or in a way, you may say a neuron sees through the proteins. That also is a way of putting it. But anyway, so protocadherins are coming up as very important targets, which also are important for neurodevelopment. But this is something which I want to finish with. The third family, we still haven't discovered, worked out in detail. Now, you see, there were 70 variations which were inherited as a block. But we looked at all of them, and what do we see? Homo sapiens, Homo neanderthalensis, pan troglodytes is a chimpanzee, gorilla, bonobo. In fact, you can have a Denisovan man also here. We also have the sequencer. Now, what do you see? A is present in normal readers. It is this, and it is different from the conserved C, which is present in the dyslexics. So that people who have dyslexia in this family, not in all families, in this particular family, people who have dyslexia have a lot of allele sharing with the more, what is known as the ancestral haplotype. The C is present in Neanderthal to mouse. The A is present in modern humans. So this sequence is predominant, this base is predominant in preponderant, as they say, in normal readers, whereas the C is preponderant in the readers who have dyslexia in this family. The same for G and A, the same for T and C here. And these two are mutations which change actually the protein and its function. So they change an amino acid in the protein. This isoleucine to leucine, here is alanine to threonine. This one affects the RNA, as we've seen in simulation studies. Here you have A and A is there in Neanderthal, but not in the others. And here you have G, which is there in Homo sapiens, but C is there in Neanderthalis and others. And these are the others, G to C, A to G, and so on. So what does it tell us? It tells us not so much about reading so as per se, because I feel still that reading the cultural adaptation. So this evolution took place not to for increased cognitive enhancement, which enabled us to read. And so we can also get evolutionary highlights when we study such families. So the the take-home lessons are perhaps this is a complex phenotype and endpoint manifestations in a broad sense are common, though we can actually fine-tune differences between them, but we are really looking at that. What dyslexia is also doing is affecting the ability of um, uh, people to read similarly in different languages. The nature of the language does not matter so much. If you're dyslexic in one language, it is very likely that you will be dyslexic in another language. So the process of reading points to a fundamental cognitive process of looking at a word, understanding the symbols, correlating it with word and meaning. And we can use genetic studies. See, endogamous groups are very common to India. It is said that there is a group of, large caste groups within India, which number more than in millions and 10 millions, have a very high level of endogamy, which can be used like at least for this generation, before people, tend to marry beyond caste, as a kind of a genetic powerhouse to study different predisposing genes. And with this study of different predisposing genes come not so much as a diagnostic genetic tool, because within dyslexia, none of these is such a complex heterogeneous disease. None of these can actually, you can put it up as a test like you can put it up for sickle cell anemia. But they will give you wonderful insights into how the brain develops, what are the cognitive processes, and maybe tomorrow we will be able to also either retrain the brain or get an early diagnosis, because early diagnosis is also, the quicker you start remediation, the better it is. The question about dyslexia is that dyslexia is it is very amenable to remediation. It takes hard work from parents and teachers, but finally the results are good, and if you start early, the results are even better. So we can perhaps understand and dissect the pathways which gives rise to neuronal aberrations during development, which lead to dyslexia, and then perhaps tomorrow we'll have a handle on the disease. Today it helps us to understand brain development. So with this, I will give you this, Bharat Prajapati, Shubha Devas is a PhD student. He had the first family to study. Shubha is a postdoc who had this family with co-inheritance of ADHD. Tista studied the other family, which had protocadherins. So these are the PhD students and postdocs who spearheaded it. And these are the people who have come and have, associated at different parts of the study, different things. Apart from that, we study long non-coding RNA and stem cell differentiation. Also, for sequencing, and there were three of us who were collaborating, myself, Nandini, and Pankaj. So these are three labs, and without us getting together, wouldn't have happened. Genomics and integrative biology, Mitali Mukherjee and Mohammad Farooq's lab and their students. RCB helped us in, especially Deepthi Jain helped us in modeling studies. Maharashtra Dyslexia Association and Masarat Khan have been very proactive. You see, most of the acknowledgement should go to the patients. It is very difficult sometimes to seek out elders in the family, try to see how they have read, what they have done, taken histories. There's a stigma attached to dyslexia, and somehow people have to be very enlightened in order to let you study them, so to say. So really, my heartfelt thanks to the families, and these are the different people from those families who have really helped us in the process. Pratap mostly, Saurabh Dalal helped us not with the families but so much in getting community-based studies, Shipra Vaidya and organizations, and NBRC for facilities and funding, and my JC Bose fellowship. So thank you very much. I hope I haven't been too long, but there we are. I'm happy to answer any questions or interact with you.
Yeah, thank you, Professor Sinha, for a stimulating talk. It was mesmerizing actually to learn so many things about, such a
Echo
complex
going
heterogeneous
on.
disease. Uh,
Somebody's echo.
yeah. So I think Professor Ram's microphone is having some echo, probably. Yeah. So it was fascinating actually to learn so many things about such a complex and heterogeneous disease. And normally we normally think that language is kind of a cultural evolution, but it was actually, I mean, fabulous to see that it's also a kind of like a cognitive evolution, and then brain plays such an important role. So the floor is now open to questions. We can also have some questions from YouTube live also. And please type in your questions in the chat box if you have any questions, so that we can pass it on to our speaker, the Professor Sinha today. Yeah, Professor Ram, actually I muted you, but you can unmute and speak actually.
So, okay. Subrata, I was really interested in this connection between multilingualism and other kinds of cognitive advantages. There are some studies which show that Alzheimer's, there's a sort of a late onset of Alzheimer's for people who are multilingual. I mean, the questions of, what are the connections between multilingualism and dyslexia, and does it always happen in all languages that a person is multilingual in? I mean, I don't know. The question is not very well formulated, but you get the drift of what I'm asking.
Yeah, I got the drift of it. Oh God, I'm muted. I hope I'm not echoing. Yeah. Okay. So the thing is this, that let me tell you what little I know about it. What little I know about it is this, that definitely there are studies from Alzheimer's disease especially, that even in, this is a work done from Osmania by Suvarna Aladi, who said that there is, even in illiterates who are bilingual, because she studied people who know Deccani Urdu as well as Telugu, no English, nothing, illiterate. There is still a advantage in the later onset of Alzheimer's disease. This is perhaps related to something which very loosely can be called as cognitive reserve. You get more connectivity and more practice. It's something like the brain being a machine. You keep exercising the brain in order to, so that by the time, see, how are these measured? A person may have reached 50% of normal, but if the 100% is very high, by the time it comes to a level by which they find it difficult to function in society, you need a greater degree of fall. And that is one way of looking at it. I'm purely talking in a very, very speculative manner. We have no clue, absolutely no clue of the molecular mechanisms involved. Molecular mechanisms is, when you do it, you need to really box up your subjects. So within families, within dyslexia, yes, these are different pathways by which the brain could be predisposed towards dyslexia. Okay. That's beside the point. The other thing you asked was if you are dyslexic in one language, are you likely to be dyslexic in another language? Now, if we take really a dyslexia which has a biological cause, the answer is yeah, probably yes, because the fMRI studies showed, see, all of these families that we have studied are bilingual families. Their scores for Hindi and their English are the same in all the three families. Okay. However, there are I frankly feel that yes, those connectivity issues are going to remain the same for all languages in a very crude way. Now, if you look at the three different kinds of scripts, English, continuous, no matra, nothing. You know, like national is brain, BRAIN, brain. Now in Hindi, you will say ba, ra, bra, and put an a ka matra on it. So on the brain, BRAIN, you have put in three matras, and na is na. So that the Hindi is read differently. Now Chinese orthography, as you all know, is different. So there's a lot of studies on people who have, whether these cross assessments work. For Hindi and English, in many cases, they do work. I'm sure there will be many cases when we go into what I call deep phenotyping. That means you try to, one is you have a dyslexia test. That itself is quite exhausting. When you, when a cognitive biologist wants to do deep phenotyping, very difficult to catch people and make them go through those tests. So I don't know how it will work out when you dissect out the finer details. At the cruder levels, I'm inclined to think, yes, if you're dyslexic in one language, you're likely to be dyslexic in the other. The other is that do you gain executive function if you learn two languages? The general consensus in the field seems yes, whether it is Alzheimer's or whether it is gain of executive function. So I don't know how I have been able to respond, but in general, that's what I know.
It seems that it should be easy, also, or not easy, but it would be something that could be verified or tested, and this is a good place to do it.
No. There are a lot of work going on in multilingual communities. You know, Hispanic versus and English in USA, uh, Spain. There's some work. Arab world, Africans are fluent in multiple languages. There's a huge amount of literature. It's just an exponential growth of literature going on. But yes.
Okay. Okay. So there is a question from Saurav. So there are many potential candidates, dyslexia genes, DYXIC1, and some ROBO1, etc.
Yeah.
You mentioned about BASP1. So we have very limited understanding about role of specific genes. I would like to know how much is the contribution from epigenetics, how much we can revert by training, education after early stage detection. This is the question.
See, two, three important issues. One is, in a way, let me say it's definite possible, and definitely there is a contribution of epigenetics. I am not really aware of the brain epigenetics and how it changes during learning or neuronal plasticity. You know, that is itself a major area for study because all learning memory is associated with RNA. RNA is the store for memory, how memory is important in decoding, how cognition is important. So those are areas, I don't have specific knowledge. Only thing I can say as a generalizer is that yes, it is very likely to be important for the very simple reason that you can remedy if you start early enough. you can do dyslexia remediation. That is the reason many of these families, especially one family was very keen where they had that autosomal dominant disorder, that there's a child who's 2 years old. Can you do the testing for us so that we can start him or her in an early remediation? Now, ethically, we cannot give out that data. But the point is, from their point of view, yes, the earlier they start, and there's another body of work in educational psychology about how to teach dyslexic children better. So all that is possible. The short answer is remediation definitely possible. How much epigenetics is there? I mean, logic and common sense says yes, but if you ask me about pathways, I mean, if there is any literature, I don't know about it. I don't know that much area in that much of detail.
Okay. Okay, so I have a couple of questions actually. I was curious about something. So you said that, of course, like it was nice to see the connection between the phenotype of dyslexia and also how it affects neuronal differentiation. So you also talked about the effect of cadherins, right? Like in case of dyslexia. So can a kind of therapeutic approach can be developed? Can it be developed for treating dyslexia, knowing that it kind of affects the cadherins, for example?
See, I'll
Or
tell you where the problem arises with the brain. The
Mhm.
brain connections are already formed.
Okay.
So you need to, whatever you do, especially neurodevelopmental, you need to do something at a stage where the development is going on. And that is true. I mean, none of us have yet worked out how to intervene at that level. So one is the connections. If the wrong connections form, they have formed, they won't reform and grow back. There are brain stem cells, but they are not so sufficient and so dynamic as to redo. So I don't know. Therapeutics of dyslexia will remain early diagnosis and remediation for a long time. I mean, I can't imagine how to an adult who has already got the wrong connections. How can you? I mean, one is of course increase plasticity, change it, directionalize it, but it's like too much of a black box, to throw things in. So I don't know where you get the specificity from.
So is it also
So
true
there
for
is one gene, let me tell you,
uh-huh.
not dyslexia,
Okay.
but in autism. In
Mhm.
Rett syndrome, there's this MACP1 gene, and apparently you can even there intervene, so to say, and get improvement of phenotype also. And so that possibility is there, but rationally, that is a single gene disorder. Rationally, how you will proceed, what steps will you take, is something that is still very unclear. We await further knowledge.
Okay. So the same thing probably can be said about the gene therapy techniques, such as like CRISPR-Cas based techniques, for example.
See,
Uh, or
possibly it is true for anything where you intervene in the brain. I mean,
okay. Okay.
in a gross sense, if you want to restore the neuronal pathways to, let us say, a stroke,
Hmm.
thick, heavy motor pathways, want to induce, people are using transmagnetic stimulation, which is known to affect epigenetics also, in order to increase brain plasticity and affect, after lesions and stroke. So, gross ways, yes, dyslexia is a quantitative issue. It's not that the person is otherwise having a problem. It's a specific difficulty in a local kind of cognitive pathway. So, I mean, it's an unsatisfactory answer, but then that's the best we can give.
No, no, I think it was insightful, definitely. So I was also just wondering, so you might be knowing about this 23andMe gene profiling, right? Which like many people in the US, almost everyone is now doing. They're sending their DNA samples and getting their, DNA sequenced and all. So I had also done that like a couple of years back. And one thing I noticed was they also give a report which says that you have this much percent of Neanderthal DNA. Okay, so is there any relevance to understand like this kind of lineage kind of genetic phenotype? Lineage analysis, what you were speaking about. So is there a significance there about understanding how much
See,
of?
the fact is, I don't know.
Okay.
It is a fact. I mean, I know that our proportion of Neanderthal DNA and Denisovan DNA varies from area to area.
Mhm.
So Tibetans and Han Chinese have a higher level of Denisovan DNA. Some people in Central Europe have a higher proportion of Neanderthal DNA. So
Okay.
somewhere, I mean, let's link it to the fact that, I mean, if we take it simplistically, modern human beings came out of the Horn of Africa. All of us are descended from one lady who they call a mitochondrial Eve, who lived about somewhere around 40 to 60,000 years ago.
Okay.
And then these people spread, and then they outcompeted the Neanderthals and the Denisovans. I mean, this is the current, and there was interbreeding, and so on and so forth. So this is the current, let us say, believed evolutionary paradigm of modern human beings. It has been known that some Neanderthal DNA genes are also associated with certain disorders like schizophrenia and so on and so forth. There are certainly with COVID, there was this issue that Neanderthals have people who have some Neanderthal alleles in certain genes are more prone to cytokine storms in COVID. Because maybe the Neanderthal, see, the point is, if you look at it, now we are going into pure speculation. Okay, human beings have died on an average at the age of 40, by the age of 40, till about 100 years ago. So the genes which were responsible for keeping them alive till 40 and giving out a second generation are very different from the genes which enable us to survive till 80 with the help of various doctors, polypharmacy, anti-diabetics, stents, of artificial knees, and so on and so forth. So evolution hasn't really adapted itself so far to all of this. And it's possible that those genes which were successful in that environment are not so useful in the modern environment. I mean, this is all, it's basically speculation. So, but that's what it is. So this is the role of the, I don't know what to make of a Neanderthal in X percentage and how it will affect my health. In fact, I feel those are very dangerous. Suppose you say that you have a twice as much higher chance of getting diabetes, but 0.75 as much chance of getting schizophrenia versus 1.7 times risk of this cancer and 0.7 times risk of blood cancer. I mean, what do? You feel happy, sad? Do you go and get yourself tested every day? I mean, how do you react to that? I have no clue. Unless something is really sharp and actionable. That is why for genetics, uncounseled genetic diagnosis is dangerous.
Okay.
That's all.
Okay. Okay.
Okay.
So, any more questions from anyone? Uh, okay, so I think it was really insightful. Like we kind of learned a lot about it. So it was a very different kind of lecture, although in our
Yes,
series,
I
I
know
would say.
from what I am
Yeah,
would be hearing,
yeah.
but anyway, that's me.
No, but it was really nice, and thank you again for agreeing to come and give a lecture
So,
on this platform.
thank you
And
so much for calling me.
And also, like, we would like to actually host you someday physically when all this COVID crisis gets over.
Definitely can come over. Yes,
Sure.
I
And
definitely love to come over.
yeah, and also I'd like to acknowledge our HOD, Professor Anil Elias, and of course, like Professor Ram, who is our mentor and a very strong guiding force behind organizing this webinar series. And also my colleagues, Professor Vishwarup and Professor Janaki, for co-organizing this with me. So thank you again,
Yes, sir.
Professor
Thank you.
Sinha. So we can conclude
Sure.
our webinar here.
Yeah.
And thank you so much. Thank you.
Thank you. It's been so nice speaking in this webinar.
Thank you.